Hello, and welcome to Quite Curious. I'm your host, Ariel Frame, and today I'm quite curious to speak with Mikko Juusola. Welcome, Mikko.
Thank you. Very, very happy to be here, Ariel.
I'm glad that you're here too. To start us off, can you tell us what your position is at University of Sheffield?
Okay. So I'm a professor of systems neuroscience. So I'm interested in how the brain as a complex system is making sense of the world. And to kind of progress this field, we are using a very broad COM. We are looking for multiple sources of experimental evidence and theoretical modeling that we try to integrate into some sort of more coherent understanding what the brain might be doing.
Yes, I've seen your team. To be forthright, I see you often because we're connected offices. Yes. So it's quite cool to be speaking with you and hearing a little bit more details of the kind of work you're doing. I know I've seen a lot of computer stuff going on, a lot of big devices in your lab.
Yes.
So excited to hear about your new paper.
Okay.
Synaptic high frequency jumping synchronizes vision to high speed behavior. So there's a mouthful there, but a lot of really cool findings in that study.
Yes.
So do you want to give us a brief introduction of what did you do in this study?
I don't know if this is brief, but this is a very long history for this paper. When I was a little guy, say that I was like 8 or 9, my teacher in the elementary school told us that the brain is the most wonderful thing, but nobody knows how it works. And I thought, okay, I'll start looking into this. And I went to library and I brought lots of books about the brain and brain function. And my first kind of a public little talk, I think I was around 10, when the others were talking about what did they do during the summer holidays, was about synaptic transmission. And I in that talk, and I've been kind of looking at the literature. One thing that I was shocked about was that this information from the environment, when it goes to the nervous system, it takes time. So that when we are reacting to something, it's not happening in the environment, but it already happened. So I almost felt like there's a castration anxiety there. That, I'm living in a delayed world.
It's wild. It's wild to think.
No. I was shocked, and now after these many years doing neuroscience, this was the question that we were addressing in that paper. So how do you speed up the processing in the nervous system such that things what happen in the environment would more or less give a synchronous representation in the brain? And this paper itself took a long time to do, so it was like 8 years work building on previous work that we've done, and so it's gradually was getting clearer and clearer for us that what might be happening. And we had then these interesting observations that kind of helped us to understand that these delays that would happen at the level of synapses, in reality, in a behaving animals, they're actually massively shorter than what was believed in the literature. And that this motion that the animals themselves are generating is actually part of the code. So the behaviors and the processing, they happen very much in synchrony. And that allows then animals to react nearly instantaneously. There's some delays, like in a matter of milliseconds into the environmental stimulus. And it also, these kind of ideas that we show here happening in the brain tissue could explain partly how you can integrate these kind of concepts simultaneously. How do you do this binding problem? How do you, when there's multiple features in your thoughts, how do you can bring them together across your brain when the distances between these neurons are sometimes massive? So I think it is a very fundamental paper, and it really happened gradually.
So that sounds like the starting point here is, baby Mikko had this epiphany that we're not living in the current present at any point. Our consciousness doesn't line up exactly. There's a delay between what we're sensing and like when we're actually experiencing that thing. And you talk about, you know, animals and brains, not necessarily just thinking about humans. We're not the only ones with brains, right? So tell us, you know, what animal did you use?
Yeah, so we have made this deliberate choice of looking for simple brains. So we typically work with insects, and Drosophila is the model system.
That's fruit flies, right?
These are the fruit flies in most of our research. But in this particularly, in this paper, we did use some Drosophila data and experiments and a bit of a modeling. But we mostly were looking at houseflies. This is a Musca domestica, which
great name.
Yeah, it is. Yeah, it's a very domestic fly, and you must have been bothered by those flies. They're just buzzing around when you're in the kitchen and you want to eat, and they want to have a share of that. And they're very difficult to catch or kill because they react so fast. And
they're much bigger.
Yeah, than Drosophila in that sense too. And we built a system where we were able to do multiple experiments on these animals. And then ultimately, after many years, put it all together into this paper. And so we looked behavior, how fast they are reacting to looming stimulus that might be a sign of threat, and how rapidly the photoreceptors and the interneurons after them are processing this information when we were changing the patterns of light stimulus, which were more naturalistic or very similar to these kind of movements that these animals are doing in the real. So animals.
So that's key, right? Because, Because, you know, you've been thought about this for a very long time. You're not taking it lightly. You're going to this mall. You didn't just randomly choose an animal and randomly choose the paradigm. You really thought through what the paradigm was going to be.
Yeah.
And you're now thinking about a fly that has to, I mean, if you're thinking about a delay, your life's on the line. And the hand's coming to swat.
Yeah.
So this fly had to, and they swat very well.
Yeah.
So and usually they do what with stimulus, and what did you do differently?
Yeah, so there's been these kind of ideas that are making big waves in the neurosciences, and they often, you know, there's a dead end. And one of these was the saccadic eye movements. Many people believe that the saccades, these eye movements, are so fast that if you had a kind of a stationary camera and you just move it very rapidly as eye is moving, then everything would be blurred. You would get this image blur.
Is that motion blur?
That's a motion blur coming from that, and this would be so detrimental for the system that it wouldn't be able to collect information about the physical reality. And so it was thought that during these fast saccades, the brain was not collecting information. And it was only during the fixations, when the eyes are still, or assume relatively still, that the information would be sampled. And I just thought that this is absolute bullshit, that the systems don't work like that. And so we...
Yeah, I guess I imagine you thinking, hey, wait a second. If you're telling me this fly is experiencing motion blur at all the time, why isn't it blind? Like, how does it see anything?
Exactly. How can it avoid my SWAT when I'm going after it? So the flies and animals in general that has vision, not all, but with the faster vision, they move saccadically, and so do flies. So
that's the like quick left right.
So they're basically doing these kind of super fast turns, and the saccades are differently defined such that the velocity is changing in like 100 degrees per second, and so this is a typical threshold taken, and faster than that is going to be considered
quick movements. Saccades are quick movements, and that can include like the eyes themselves moving in our case. But that can also refer to like the head moving. Or the body moving.
That's right.
And then also what you found.
The photoreceptors also, these light sensors we have earlier found that they are photomechanical. So if there's a light change, their structure changes, so they're physically moving.
So that's kind of wild in of itself. Like I mean, we know our eyes move left and right, and when we have a very different eye than a fly, right? So now we get a picture that we have this fly. It's got a bunch of actually different eyes. We call them ommatidia, right? And each one is doing its own little.
That's right.
Like a piston, in, out.
That's right.
And maybe left, right too, or turning.
That's right. So there's a very beautiful organization across the eye. So the left and right eye, these microsaccadic movements inside ommatidia, like you said, they are mirror symmetric.
Microsaccadic.
We call them microsaccades or photomechanical microsaccades. So when they are happening, they happen so fast that you can't see them by naked eye. So we had to build this kind of a high-resolution camera systems and use infrared imaging. So because if you put infrared, the fly doesn't detect that. So we can detect these movements, and the fly is oblivious that we are looking inside their eyes. And so that allowed us to see that there is this constant communication happening. And each time, of course, when the photoreceptor is moving or changing its position, then the information, what it's collecting from the environment is changing. So it's kind of auto-regulating what's falling into its receptive field by doing these changes. And like you said, it's like a piston motion when these cells are doing. So the light increment is causing a contraction. And so the cells are moving away from the lens, so then physically they are collecting light from a narrower angle. At the same time, they are moving in one specific direction, which is set by development across the eyes. So it's going to, like a in the car's engine, the cylinders are having the motion inside them that is very specific, and so it's like a piston motion when the piston is moving. And that's
wild. And you discovered that happens at all.
Yeah,
in the fly eye, and you found that happens. It happens in the fruit flies as well.
Yeah, it happens everywhere. So even in your eyes, it's been since found that the photoreceptors, the cones and the rods, what are our photoreceptors, they are also photomechanical.
And they do microsaccades.
Yeah, they don't call them microsaccades, but they are microsaccades. So they have a very similar waveform.
What?
So after we published our work, there was a work from Stanford. Some people used this kind of fancy ophthalmoscopes that allowed them to see directly to the fovea and foveola through the lens. So they could see when they gave a light stimulus through that system that what's happening in a fast time frame inside these single cells, and they found out that they're physically moving. So light change makes them move. Then second paper came. There were people from Trieste. There was Vincent Torre and Trevor Lamb were involved in this. I think they use Xenopus photoreceptors and rods that they had dissociated in a petri dish, and they were flashing them with light, and they moved the same way as what we see in the flies, except it's a bit slower in our case. But basically the dynamic of contracting and elongating was very similar.
Okay, so now you discovered at some point that this is happening.
Yeah.
And in this paper, you're kind of working out why.
Yeah. So we had ideas why. We had a few papers, but we didn't know how this is related to the information transfer further to the brain. So we was pretty much establishing this dynamics more at the level of the sensory surface, but then how is this communicated through the synapses, and how this information is then changing how the brain works. And so in this paper, one reason why using these bigger flies was that we had earlier developed the methods where we can intracellularly, by using this kind of conventional very sharp microelectrodes, to look at these electrical responses that are induced by light and then induced by the synaptic transmission from the photoreceptors to these first order interneurons. And we wanted to see what happens if we give light patterns which resemble those light patterns that the fly eye is normally experiencing when it's moving saccadically in the environment. And so we call these a bursty light stimuli. So when you move eyes, you usually fixate on a darker objects. This seems to be happening. So even when a child is born and it's looking at the mother, it finds these two dark dots, two eyes and a mouth, and it's just rotating. So it fixates on those, and then it jumps over them through the nose and forehead, or you name it. And it does this very cleverly. And so we had earlier worked that the information sampling from the environment by these photoreceptors is following this kind of, we call it stochastic rules, stochastic refractory rules. And so they are kind of samplers or sampling machines that are evolutionarily tuned to enhance certain type of patterns when things are changing in the world. And the animal behavior, the eye movements, or the microsaccades themselves, partly contribute to that. So that it makes it more it makes it easier for the animal to actually resolve things happening in the world when they are moving in these certain ways, because of the sampling is this kind of stochastic refractory. And I could go into details, but I don't think the audience would be following me. But it seems to be happening everywhere, and we are now using the same rules when we are modeling what happens in the synapse. It seems to be using the same thing. So everything is basically based on the idea that the information in this world, as we experience through our sensors, is quantal. So there are photons which are falling onto these cells, and there are these light-sensitive rhodopsin photopigments that are then reacting and doing conformational change, which then leads to a cascade of events. And ultimately, there are ion channels on the cell membranes that are opening, and at the same time, or just before it, the cells are actually contracting and or elongating, depending on if it's a light increment.
So let's orient here to the experiment, right?
Yeah.
So you've got this electrode, which is really like a tube of glass that you've sharpened so sharp that the one end is so narrow that you can stab it very, very lightly inside of a cell.
That's
and,
right.
And measure what is actually inside of there. And
that's
really,
right.
when you're talking about ions moving here, that's like electrical.
Yeah.
That's what you're actually measuring.
That's
electrical conductivity inside of a single neuron.
That's right.
Neuron.
That's right.
And this is a particular neuron in the eye of the fly.
That's right. So when I was a research fellow in Cambridge many years ago, I took a risk because people were saying that you can't do intracellular recordings from Drosophila, these fruit flies, because they're so small that it's impossible. And I thought, well, we'll see it. And I spent like 1.5 years of building up a system, writing also a very big software package that allowed me to do things very rapidly with these instruments and
like electrophysiology.
Electrophysiology.
So this technique is notoriously difficult
so
for
now,
anybody really. Neuroscience always neuroscientists think about this technique as a particularly difficult one. So you took on a great challenge.
That's right. So the benefit what I had in Cambridge, this was in the physiological laboratory, was that they had super good workshops. So and I only had to pay for material. So I basically used over 80% of their resources just building stuff with these super clever guys. And so we built all sorts of stimulators, and we built those mechanical systems that were so stable that we could then drive these electrodes very nicely.
Yeah, because the movement is going to be the issue, and
it's a
perpetually
killer.
you lose the cell.
And
the cells are small.
Also, the head of a Drosophila is like a few hundred microns in whole, and you have to remove like 1 or 2 ommatidia size of things on the cornea in order to drive these electrodes. So there was a lot of very good challenges to take. So people didn't believe in the beginning because the quality of the recordings was so good, so that we could use these kind of methods like information theory to look at how the light information is then converted into the electrical information, and still people don't really believe. So we made videos and made this kind of a Journal of Visual Experiments articles so that the people actually can see. And in one point, we even were doing an EMBO workshop so that the students could try it themselves and see that it actually works. Yeah, so that took some time, and we really kind of almost like perfected it to get really high quality data, which now allows us to, when we use a bigger insects in the same setups, we can keep these cells for hours.
Yeah, I mean, after working with Drosophila, fruit flies that are way like 10 times smaller.
Yes,
like much smaller.
Yeah.
Now working with Musca domestica, the housefly.
Yeah.
I mean, now it's a luxury.
Yeah, no, it is. So it was kind of easy. So we had published a paper earlier in a journal called eLife about these microsaccades and how these kind of saccadic movements are maximizing the photoreceptors' information take, which we just showed that if you move these cells in certain ways, then this is the most efficient way of collecting information from the environment. And now we just wanted to do this into these interneurons that are notoriously small in Drosophila, and we can do recordings from those, but they are very rare, and we can't keep the cells so long. Typically, I think the longest recordings we ever had is like 30 minutes, and after that, the recording quality, and even during that period, the recording quality is a bit suspicious, or can be suspicious. But if we use bigger flies, we have like superb recordings from these interneurons, and we can repeatedly do these recordings, and And because we wanted to test such a large gamut of different stimuli just to show that these particular type of patterns with certain frequencies or burstiness.
And not to make the comparison directly, but I know that there's a lot of vision work in the past in like cats. I don't know how good the quality of the recordings were. I'm not an electrophysiologist. Are we talking like good with the flies? It is good as you'd expect to get in an animal like a cat, or is it comparable?
The science is always limited by the instruments, and we also kind of infer the scientific knowledge through instruments, and that kind of biases our understanding of them. And this is true. So in the early stages, they had this kind of amplifiers that took a long time to build, and they used all tube structures rather than transistors to get going. And Hodgkin and Huxley and others, they were able to then get recordings from these giant squid axons that were so big that you could put a bar of metal inside, so that you could measure the axon potentials. And so those were high quality recordings. But then the recording systems were very elementary. So you had oscilloscopes, and you had to take pictures by using classic film.
Yeah, we're talking an oscilloscope is like imagine a screen next to the bed of a patient in a hospital where it has a little line going beep 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 beep, and that is being recorded by like a pin on a sheet, like printing it out, and then you take a picture of it, right? Yeah. And now it's all like in computers. So
that's right. So the modern computers allow you to get data in a very high frame rate and digitization. These are 16-bit channels sometimes, so you get a very high resolution for the voltage patterns that you are recording. And so it's better, but of course there's lots of assumptions also in these recordings, because you put the electrode inside the cell, and you call this recording as an isopotential. That means that everything inside the cell would be in the same voltage, but the cells have many shapes, and there could be local computations that are in a completely different voltage than the voltage what you are recording at one point in space. So there's this kind of abstractions and assumptions that are reductionistically giving us some tools to look at what might be happening inside the cell, but it's not the reality. So we are always limited by the instrumentation, and always these experiments gives us a rather limited view, because the animal is restrained, like the fly. We fix it so that it can't jump or do anything else there, so it's like in a straitjacket, poor fellow. And we put this tiny glass electrode in its eye, and we collect that. I'm sure it's not happy. So the responses what we get might be also reflecting its unhappiness, because I'm sure there's a lot of top-down signals that it's sending to the eye, and the body parts it wants to escape, but it can't. So we assume that everything is stationary, that we get these specific recordings, and we analyze them, and that would be the quality of signaling what happens in the neurons. No, it's an underestimate. But even so, these techniques we built and modified to get these recordings, they are really the top of the art. So we get such a high quality signals that the signal-to-noise ratio of these neural patterns when we are repeating can be several thousands.
That is wild.
That is wild.
Like, I mean, a lot of people are aiming to get one or two.
Yeah.
Right?
So
that's just to say that, like, there's some amount of noise. Imagine like static on a screen, and you see there might be patterns somewhere. The static on the screen would be noise, and how clear the pattern is how much signal there is to the noise. And if you can just barely work it out, then that might be like one or two. Yours is like the static is very obviously different.
That's right. So we
pattern.
invested lots of time to grounding this system so that there's a very little instrumental noise. There's always an instrumental noise, 1/f noise, but these are as low as we can go. But in the neurons, we now are quite convinced that there's very little noise, whereas the current neuroscience is saying that the brain is noise, that it's you need lots of samples to get through that noise. And when we do these recordings in individual cells, and also many people are believing that the neurons work as filters, and we are completely trying to transform this to that these are these are stochastic systems where the form and the functions are very tightly integrated, and that when they are operating and communicating with each other, there's hardly any noise. And that this, what we call noise, typically is a processing which we don't understand where it's coming from, because the neuron is simultaneously communicating with many neurons. And we think that there's only one source of information going through the system, which would be the light stimulus. But at the same time, the other neuron might be whispering the one that is looking at the light something else, and now we say, oh, this is noise. No, it's just integrating the previous history of knowledge, what it has, or some sort of it could be some clock systems that are synchronizing, and it's coming through, and it's part of the processing.
Yeah, if you look at a complicated system, and you look at one measure, and you like summarize the average of a bunch of measures, if it's accounting for many, many different things, then anything that's not exactly the output of the exact thing that you think it is might look to you like noise.
That's
but
right.
It's actually accounting for many different things going
That's
on
right.
at the same time.
And so there's the assumptions have been, in my view, massively biased our views of seeing the nervous system, because
what are those assumptions?
There's like this, that the encoding is noisy, and that also that the action potentials are the only form of information that the neurons are using.
So action potentials being
like
when every neuron they fire, and it's
so
like they send a signal, and it's like on, and then it turns off. So the assumption before was it was only on or off.
That's right. So it's so-called digital, so that the people say that the information processing in the brain is digital, and so when you look into this deeper and deeper, you find that it's not. When you're analyzing these action potential patterns, when you have a high resolution recording system and you have very low noise, you find that if you give a same kind of stimulus pattern, you get action potential patterns which are unique for that stimulus pattern. Their waveforms are unique. So if you have in the cortical neurons, as an example, we published a paper 20 years ago. So when we were looking at if you had naturalistic conductance patterns, this was in rat cortex, and we found that if you have a pattern, we call them NMDA or AMPA type of glutaminergic inputs that we were generating. These conductances were similar that we were injecting into these cells, and we were looking at the action potential patterns that the neurons are producing. And we found that if there's a high conductance state, so if you have a patterns of lots of activity and then something going on top of that, and if it triggers an action potential, you get, we call a kind of a short and a fat spike. And then if you have a lower conductance state and you have a suddenly changing input levels in these conductances, you get a tall and narrow spike. And so now by clustering the spikes to tall and narrow spike and looking at the stimulus history, we could predict the stimulus directly from the action potential waveforms about 50 millivolt before the action potential waveform happened.
Yeah,
so there was
based on
so there's no noise.
Acceleration of
there's
the
no
curve,
noise. And now this is seen in multiple preparations. So people have not really been actively publishing or saying that this is what they see. They look at, we can follow the rates up to 1 kilohertz in some systems. But if you look at the patterns of spikes that are happening and you give up that idea of digital 0 and 1s, you actually see that the action potential waveform is carrying information. So we wrote some review paper and done some other work many years ago, and there was hardly any reaction to that. But we were pretty much predicting also that through the synapses, this kind of patterns, if you have a tall and narrow spike, you would get a different kind of EPSP, which is kind of excitatory synaptic potential on the next neuron, how that information is conveyed through the synapse, than if you had a tall and fat spikes. Short and fat spike. And indeed, this then was later shown in a Nature paper that if you were able to patch clamp on the PLEP, just on the presynaptic site before the synapse, and then on the postsynaptic neuron, those conductance stages, if you were in a high depolarization state, which is typical for short and fat spikes, you get a bigger EPSP on the postsynaptic site.
So just to understand what you're saying to get to this point that it's not just on or off, it's not just digital, it might be analog.
It's
that you found an instance where you can measure the one neuron speaking to the next neuron and measure the one side and the other side simultaneously. And on the one side, when it's speaking, you're measuring on and off, but like different shapes in which they were on and off, and actually changed. How
we didn't do these recordings ourselves. It was a group in America, McCormick group.
But they came predicted this would occur.
We predicted this, and we tried to publish in Nature. We we had this action potential waveform code, and then there was a lot of resistance from the referees, and of course it didn't come through. And then eventually we published it in the Journal of Neuroscience, and it's kind of funny. So the paper came out there, and I got one email from some guy, and the guy was, "Oh, this is a great paper, good to see it out." And the name of the guy was Grigory Buzaki, and I didn't know who Grigory Buzaki is. I wasn't sort of reading this field at the time, and I showed it to my friend Hugh Robinson. I said, "Hey, there's some grad student who was interested at least in my paper." And I said, "What? This is from Grigory Buzaki, the famous Buzaki likes our paper." Yeah, there were some people who reacted, but overall, it was ignored.
I mean, it can be slow, right?
Like, yeah, it's slow. No, it's fine. But
can we relate this this idea that you've had for quite a while and it's been recognized by folks to the findings of this paper and what you found at this photoreceptors?
Yeah, so it came even sexier than what we thought. So when we were driving these early visual systems in fly by using these kind of bursty patterns that are characteristic for moving rapidly saccadically in the environment, is that the synapse is working massively faster. It was transmitting this information in a bandwidth that thought to be impossible. So if you had a stimulus that was maximally going to about 50 hertz, so relatively, so the lights in your room, if they are neon lamps, they are just following like a 50, 40 hertz, and to you it looks continuous, but it's relatively low frequency. And so the fly synapse was able to jump this information into frequencies up to 1 kilohertz.
That's wild.
It's completely crazy. And by integrating this information simultaneously from 6 photoreceptors, so there's 6 photoreceptors roughly looking at the same space.
So each ommatidia, they have like all these ommatidia, which are kind of like each having their own, they're kind of like each their own eye, sort of. And each have 6 specific photoreceptors in a special pattern. And I think people might imagine like what an eye looks like. In movies, you see this very distinct looking insect eye, and it looks like little hexagons, and that's because of those patterns of those each ommatidia.
So there's a degree of redundancy which helps to increase the signal to noise ratio of the system. So there's 6 cells roughly looking at the same point in space, and now these 6 photoreceptors, they're physically moving all slightly in different directions with the microsaccades, and then they are pushing this information to this interneuron, which is called a large monopolar cell. And in these large monopolar cells, now if you had an input pattern, say that it was 50 hertz bursts, it was going to 1 kilohertz. And
that's wild. So it's somehow like predicting it gets faster than the input.
That's
right. How is it doing that?
So we showed that we call it predictive coding, and it actually follows the laws of physics, and we are able to model it very nicely. So the whole processing goes back to this quantal information sampling. So you have n number of these kind of sampling units. We call them microvilli. And so
microvilli, these little protrusions on the side of the photoreceptors.
Yeah, they're like mini finger-like formations that are glued together. And in case of Musca housefly, there's about 40, 50, 60,000 of these together. And each one is a light-sensing unit, so each one can respond to single photon.
So you have 6 photoreceptors, each of which have like thousands of these little fingers, and each one can detect a single photon.
That's right. And in our models, we are biophysically modeling these microvilli. So in the model has like 60,000 microvilli. Wow. Each microvilli is having a full reaction cascade that we modeled by diff equations. And we learned that there are these properties called refractoriness, which is that if the one microvilli is responding to a photon, it takes some while before it actually produces this tiny voltage response. And during that time when it's producing the voltage response, it cannot respond to another photon.
So there's a period where it's detected a photon, and it's kind of like, I got to take a breather.
That's right.
Let me take a second.
Yeah.
Or so not a second, a fraction of a second.
And these refractory periods seem to be synced by the behavior, so that when you're fixating on these target patterns, then it's long enough, those fixations, that you remove this refractoriness from your microvilli system, so that when you jump your vision, like if you're a baby, from your mother's mouth to the nose, it crosses this brighter period section of the skin where there's more photons coming in, and now those photons are all available for microvilli, and they produce bigger responses. So it makes the photoreceptor responses very large and bursty, but it sequences these patterns into this kind of again, like a finger, like patterns in time that are very accurately describing what's happening in the environment. And anyway, so when these kind of inputs are then, when there's like a 6 parallel inputs coming from 6 photoreceptors, and they're all like these massive finger-like thingies, which are roughly in synchrony, coming to the synapse, then it kind of overwhelms the synapse. So synapse has a operational range, which is limited by all sorts of physical factors. And so there's a roughly about 60 millivolt response that it can handle. But now when there's 6 photoreceptors simultaneously pushing the information and having this synaptic vesicle release and the transmitter molecules going to this one neuron, the synapse operational range starts to clip. So if you had a sinusoidal pattern which is running, then all of a sudden it converts that sinusoidal pattern into a square waves. And in the physics, you may know that if you have a sinusoid and you then replace it with the square waves, you get these massive harmonics. And these harmonics are chumping that information to these high frequencies. So physically, the system is making the photoreceptor movements and this refractory sampling the signal so big that they start to clip. And when you clip, you don't have to wait that signal to come to the top value. So it saves time. So it becomes predictive. And
okay,
now,
so just to like center myself, this is pretty a little bit difficult for me to follow. So I'm going to try and translate this to a simple idea. So I'm imagining I'm running up a mountain.
Yeah.
Okay. And it's to the shape of a sinusoidal wave.
Yeah.
And the mountain goes. I can run from I'm the bottom on one side.
Yeah.
I'm running up to the top.
Yeah.
And then down to the bottom of the other.
Yeah.
But I run up halfway the mountain, and I realize I'm going down this mountain anyway. I know there's a down on the other side that's the same as the up I just did. Why don't I stop halfway and just walk along the middle?
Yeah.
And then walk down again.
That's right.
And
so
that's the clip. That's
that could be that kind of clipping happening.
So there's like you don't have to go to the whole rest of the top of the mountain, so it's faster.
Yes, that's right. And because you have these 6 photoreceptors that are slightly out of sync because they move slightly in different directions, and they are integrating these signals, it gets lots of predictive power. So they get lots of samples from the same point in space roughly that are slightly different, and this variability then is used here to increase the precision.
So the large monopolar cells, they recognize that there's these sorts of patterns. They're like it's encoded in them somehow that they know that this is the type of pattern of information that's going to be given to me. So when I see mountains, I just clip them off. I know that it's coming. So I know what's coming next. We can do this faster.
It's a bit more elaborate than that, because these monopolar cells also have a synaptic feedbacks back to the photoreceptors. So they're kind of auto-regulate.
So they receive input from the photoreceptors, and then they send something back.
So photoreceptors have basically 2 sources of information. One is from the environment and the other one is from the brain. So there's constantly finding a balance. It's kind of a balancing the synaptic loads, the inputs and outputs. I mean, they're both 2 sets of inputs, one coming from the brain, another set of inputs coming from the...
Would you say externally and internally?
Well, they both this processing in the synapse. It's happening internally.
It's all happening internally. One is one step away from outside.
But what happens there is that they use these feedbacks to constantly reset that operational range of the synapse so that this clipping is highly efficient.
Okay.
So that if the patterns of inputs are changing now, say that you have some section in the environment which looks the same, then this settles down. So there's not so much signal going on. But then again, when it immediately sees some difference, which happens when the eyes are moving, you cross an edge or something of the object, this gives a massive enhancement, and this then clips the signal because it already knows that there's something important, and it passes very fast. But the important thing here is when it's doing this, we can actually physically model it. So our models are elaborate. They are taking into account all these biophysical factors, and these been taking for 30 years to build, but they work. So these are kind of the only models I believe there is out there where the model is doing pretty much what the real cells are doing, because the models are, we call morphodynamic. They are kind of algorithmic representations of what the cell's structure and function are, rather than saying that this is a filter, that it filters the structure of the cell, now how many microvilli it is having, and how these microvilli contractions then are integrated into this whole body movement, which is then effectively regulating how the light information is falling into it dynamically, and then how the synapses and the synapse numbers, how they've been matched.
So morphodynamic, morphos like the shape, dynamic means it's changeable. So there's a shape, and the villi, and they're like changing in some way, and the large monopolar cells are morphodynamic too.
And the photoreceptors are morpho. So we're talking about morphodynamic computations. So this is very different what you see in the textbooks, because there the information processing is typically thought to be electrochemical, so that the synapses are converting these electrical changes into transmitter concentration changes, which are then picked by receptors, which then charge the postsynaptic cell to again do a
more simply, they're taking like an electrical stimulus, something electricity, turning it into like a chemical, that's the transmitter, and then back to electricity. And this takes like a certain amount of time, and it doesn't account for this morphodynamic.
So in what we call like a morphodynamic computation is that we mean that the neuroinformation processing is not only depending on this electrical signaling, but also it's precisely controlled by the movements of the cells and the changes in the form of sensory organs and neurons and synapses. So they kind of have lots of genetic information through evolution, how they should react to that. And the motion of these sensors themselves are actively determining how information is actually sampled. It's transformed, and in this paper we show that it's actually synchronized, so that if we look at those signals that are happening at the level of photoreceptors and the monopolar cells, and we had some evidence from later studies where we had used a fruit fly, and part of the data that we also showed in this paper was from the fruit fly, is that the fruit fly responses at the brain level. So these were lobular plate tangential neurons, like 4 or 5 synapses away from photoreceptors. They are in synchrony what the monopolar cells are doing. So if all these synapses were using the same kind of rule of this kind of clipping the signal in a certain way, doing this synaptic high frequency jumping, then it would synchronize the system, so that even the later cells, if they use the same rule, would be that it's like a dominoes, that if you make them to fall, all the early ideas were that the one domino at a time it falls, but now what we are saying is that these dominoes are connected by this tip link. So when one starts to move, the whole cascade moves at once, and that this is tuned by evolution to have this structure, and it was surprise to us to see. And so we tested it further by looking at the behaviors, how fast these animals can react. And we found that they have these kind of voluntary behaviors, like if there's a looming object coming on, they might react to it or not. If they might be in a mood that, this is scary, or they might not be. And when they react, they do it at a different speed, sometimes very fast, sometimes very slow. But if you look at those very fast movements that they are doing, pushing them, and sometimes they are just a bit scared and they react more likely than the other flies that you test, you find that these reaction times are pretty much matching the neural signaling. So we had the shortest delays in some of these experiments we've done is like 13 milliseconds from the point when the stimulus started to change. So it's like
almost
instantaneous,
almost 1/100th of a second.
So it's kind of a crazy, and these were voluntary, so these were not reflexes, so it's a more complex process. So there's constantly these animals are thinking, and then they use some sort of evaluation criteria why they should move and what they should do. But these movements themselves enable the system to be this fast. So and this was not considered at all in the old neuroscience, because it wanted to do this reductionist view that it's only using the electrochemical signaling and everything is stationary, nothing moves, and that it was easy for the flies because you have this exoskeleton, so it's easy to believe that everything, because it's so stiff, you don't think anything moves inside it, but inside that exoskeleton everything moves, and it's amazing that it's not been really seriously considered. Maybe because the experimentation and the modeling was not able to catch up. But now we have the tools. We can see this, and we should model it and test it.
Yeah, I know. It's the case sometimes you have these ideas, and you're just thinking, I don't know is that even possible? Can we even do that? And so you work your way over years, and now technology is advanced that like we can do it. I mean, people perpetually, I'm sure you get this. We say, hey, what do you do? Oh, I work with flies. How? How do you do that? Well, we managed quite well now.
No, and I return to the same point that even though we have these fancy instruments, we are still underestimating how well the nervous system is doing, because we are basically quantifying the instruments, what's coming through them, not the nerves. It's very complex field. We have lots of ideas how we would like to take this further, but the problem is that when you put these new ideas out there, it takes a long time before people start to accept them. The science is a bit like religion. You get stuck into some sort of imaginary world which you think is a better explanation for reality, and it's very hard to snap out of it. And now we are saying that, hey, this previous states of this imaginary world, what we have created for us is in a point that we should rectify some of the assumptions, and it's very, very hard to get the message across, and very, very hard to get funding for that, because people are not necessarily ready to react.
But this study was done really systematically, really carefully, really compellingly. This is good results. So now I think if there are people with the textbook idea that you said you'd kind of overturning a little bit, they need to reconcile with the fact that you have real results here. These flies are responding very, very quickly. And these cells are responding like almost instantaneously.
But it's also
so that is happening. You measure these cells; they're doing it very, very quickly.
But if you generalize this, and let's think about this conversation what we're doing here. So I'm you ask questions, I'm drawing some answers. You ask, I ask. It's interaction. How in the earth we can do it so fast? How in the earth, and also these words what we're putting out, they carry only very few bits of information if you take each word and break it down in a Shannon way. But then I can only say a few words and you already get the concept. So the information transfer is massively bigger. So that means that our brains are ready. So actually the most information is already in the brain, in the communication channels what we have. And we are able to react predictively to this by having just few syllables. And the thoughts are crossed, and this can be very complex thoughts. We can make these images in our head. It would be impossible to do if you had this kind of digital slow processing with delays.
So the idea here, are you saying that you've got this idea that like information can be transmitted much faster than we would have expected?
That's right.
So there's these assumptions about how fast information can be transmitted, and they don't necessarily bear out, and you now have evidence against how slow they would have expected it to be. And that can apply not just to flies seeing or zebrafish seeing or us seeing, but also to taking in information like audio, and also not even just taking that in and deciding to move, but taking that in and having a thought. So actually cognitively processing anything.
I'm completely mystified how these words come to my mouth. I don't have any kind of inner voice. All of a sudden something comes out of me. I don't know where it comes and how I'm integrating this. And I have some friends that are super good in maths or something. And I'm just amazed how they see these things. And if you were to reduce this sequence of patterns of information, this kind of a classic Shannon way of communicating, it just doesn't happen. So that means that there must be a semantic representation all the time in the brain, and that then it's already inherently in these world maps that evolution has given us through, and then the embryogenesis just like a virtual reality. It puts these systems in a place that we are ready for this world. And indeed, you see this with the animals when they are born. They don't have to learn things. They know how to things because the genes taught them when they went through the.
And there might be more that's innate. Than we
so that's a kind of a beauty with the biological AI now. So that you see the AI in the world, they're using these large language models and basically telling the language, this artificial intelligence, what we get naturally from the genes. So this information is somehow zip code and packaged in there, and then it's just unwrapping and giving us this consciousness as we then go along. And the learning is very minor. It's just a pruning of these existing connections, and it's amazing. So I think neuroscience is just taking these infant steps. We really don't understand the things, and we've been really stuck with these assumptions for a long time, and we should open our eyes sometimes.
It is
yeah,
exciting.
Yeah.
Not necessarily everyone appreciates
No,
it, but
no.
I hope that at least a few seeds are planted.
Yeah,
and
yeah.
There's some excitement out there, and there's people just like baby Mikko was excited about the prospect of discovering how the world works, how brains work, how we manage. Now you got a little bit of answers. For that question that you had about like how's it possible that there's this delay between what we're sensing in and then how we're experiencing. Are we experiencing things that are happening at the same time as we're experiencing, or is there a delay? Maybe there's a little bit less of a delay than you think.
That's right. So it's for sure that there's a delay in this, but I think it's tuned to our behavior so that we don't really experience it unless there's a bullet flying and we can't dodge it. It hits us and that's the end of us. But overall, most of the things that in our environment we are able to react quite swiftly and survive pretty well. Yeah.
Okay. Well, this paper, pretty exciting. I encourage people to go and read it, and we're going to put a link in the description. I'll say as well, you brought to my attention that mixing AI processing, and now we have a podcast. You mentioned there's this other podcast that someone used an AI to describe your paper, and they did a pretty good job. They do different.
They did a good job. Yeah,
different
I was impressed.
sort of explanation, different kind of discussion than we're having here.
Yeah.
And I'll link that in the description too if people want to have a different type of they want a computer to explain it to them, and they can, and they in some ways computers, they got their benefits. They're different than us. So yeah, I'll signpost everyone to all those things. If somebody wanted to find you online, your presence, website, something like that, anywhere they wanted to catch up with what's going on with Mikko Juusola's lab and your work, where could they go?
I think it's easier just to Google my name, and then you find our website and the other material that we put out there and the papers. Google Scholar, of course, gives direct access to the papers if you're interested. Yeah.
Okay, I'll put a link to your lab website and the Google Scholar in the description. But
Good.
It's been a great chatting with you, Mikko.
Likewise, I really enjoyed this. This is good fun. Yes.
It is great fun. Yeah, it's been great to speak with you, and we're gonna have chats.
Oh yes.
in the lab when I see you all the time. Anyway,
yes.
so thank you for coming on.
Okay, thank you.